Nucleic acids are composed of nucleotides, and nucleotides are composed of three parts: a phosphate group, a 5-carbon sugar, which is deoxyribose in DNA or ribose in RNA, and a nitrogen base. There are two classes of nitrogen bases, purines and pyrimidines. Purines have a double ring structure, while pyrimidines have a single ring structure. Note that RNA contains the pyrimidine uracil rather than thymine. And here's another diagram that shows you the structural differences between the two nucleic acids. Notice that RNA is single-stranded and contains ribose and uracil, and that DNA is double-stranded and contains deoxyribose and thymine. There are some key structural features of DNA you should be familiar with. DNA is a double-stranded double helix with a uniform diameter. The helical shape is due to the sugar phosphate backbone forming a coil on the outside of the helix. This causes the nitrogen bases to point inward toward the center. DNA is also antiparallel. This is because the phosphate groups bind to certain carbon atoms on the sugar molecules. Each carbon on a sugar molecule is numbered, as shown in the diagram above. The nitrogen base attaches to the 1' carbon, while the phosphate group attaches to the 5' end. The 3' end has a free hydroxyl group. That's the OH that you see there. This allows the phosphate group of one nucleotide to form a covalent bond called a phosphodiester bond with another nucleotide. How the nucleotides are assembled to form functional nucleic acids is this. The sugar and phosphate groups form the backbone of the molecule, which is held together by covalent bonds known as phosphodiester bonds, shown here in the pink. The nitrogen bases are connected to one another across the middle of the helix by hydrogen bonds, shown here in the green. Nucleic acids also share a characteristic called complementarity. In 1950, Erwin Chargaff, a biochemist, determined that DNA samples from multiple species all shared a few key characteristics. He found that the proportions of adenine and thymine were equal, and that the proportions of guanine to cytosine were equal. This led him to conclude that the proportions of purine nucleotides, A plus G, were equal to the proportions of pyrimidine nucleotides, T plus C. This observation led to the formulation of Chargaff's rule for base pairing in DNA, which states that a purine must pair with a pyrimidine across the double helix. DNA replication ensures continuity of hereditary information. You should know that DNA replication is semi-conservative. This means that one strand serves as the template for the formation of a new strand. This new strand is complementary to the template strand according to Chargaff's rule. DNA replication takes place in eukaryotes during the S phase of interphase during the cell cycle. And in prokaryotes, DNA replication precedes binary fission. DNA replication involves several different enzymes and protein structures. Let's talk about these as we walk through the process of DNA replication. First, DNA must unwind at what is called the origin of replication. An enzyme called DNA helicase, the blue arrow, unzips the DNA molecule, creating a replication fork. This allows for bidirectional DNA replication. An enzyme called topoisomerase, represented by the green ring, prevents DNA from supercoiling during the replication process. DNA polymerase is the enzyme responsible for adding new complementary nucleotides to the template strand, but it can only do this at the 3' end of a DNA molecule, so replication must proceed differently on the two strands. The leading strand is replicated into the replication fork, and its formation is continuous. The lagging strand is replicated away from the replication fork and is formed as a series of short fragments called Okazaki fragments. These fragments are then stitched together by DNA ligase. One of the most important things you need to understand when learning about nucleic acids is the idea of directionality. Let's use hands as a model. The carbons in a sugar molecule are numbered 1, 2, 3, 4, and 5. This matters because DNA is what we refer to as an anti-parallel molecule, meaning that one side of the molecule runs right side up, while the other side runs upside down. This is important because during DNA replication, DNA polymerase can only bind to the 3' end of the sugar molecule on the DNA nucleotides. So when DNA replicates and the replication fork is formed, DNA replication can only proceed in a certain direction on each strand. On the leading strand, DNA replication proceeds into the fork. On the lagging strand, DNA replication proceeds out away from the fork. So remember, directionality matters. Finally, it is important to remember that nucleic acids are information-bearing molecules. They contain the genetic code needed for the manufacture of proteins. The term central dogma describes how information flows in living things. This means that the information in DNA is transcribed into RNA during transcription, and then this information in RNA is translated into proteins during translation. This flow of information is true for most organisms. Genetic information is stored in and passed on to subsequent generations through DNA molecules, and in some cases, RNA molecules. Retroviruses are the exception to the central dogma. They utilize an enzyme called reverse transcriptase to produce a DNA copy from the RNA contained within their viral capsid. This DNA copy they produce using reverse transcriptase can then be integrated into their host cell's genome. I hope you've enjoyed your tour through DNA and DNA replication. In this lesson, you should have learned about the importance of directionality in nucleic acids, the process of DNA replication, and how this process differs in prokaryotes and eukaryotes. Now get ready. We're getting ready to learn some of the really tough stuff, how DNA communicates with RNA to help cells make proteins. Let's go.